immunity component, which is a very exciting part for a natural component which can target Survivin, the tumor survival factor. The modern medicine, everyone knows that it aims to have developed safe, efficacious drugs which are affordable to patients. And there are the setbacks with the conventional anti-cancer treatments, and there are the side effects, the poor oral availability of the drugs, short plasma half-life, drugs are resistant, and there is a recurrence of the drug-resistant, radio-resistant tumors, and patients' compliance also because prolonged intravenous administration of the drugs into the cancer patients is not easily accepted. Our lab has a research focus from molecular oncology to molecular cardiology and molecular neurology. You must be thinking that, but here we have a thread within all the three areas, and they have the two proteins, which one is the lactoferrin and Survivin. So our research goal is basic biomedical scientific findings, which will lead to the therapeutics and diagnostics for the translation potential in clinics. You have heard my colleague, Professor Kanwar, in the morning session, and maybe you'll find some of the overlapping with, in the introductory session. So we are working on to have the safe, efficacious drugs. With the recent fast developing field, nanotechnology, so we have doing the nanomedicine development where we are developing the nanodrug oral formulations, and which we test through the in vitro and in vivo models. And our models, our lab has a number of models: colon cancer, breast cancer, and inflammatory atherosclerosis and arthritis. So they are the different models, but because inflammation and cancer has the same mechanisms, anti-inflammatory mechanisms, and the proteins, survival proteins, so this is our focus. But today, I will talk only from the cancer perspective, and in the molecular pathogenesis and drug discovery of cancer, so these two proteins, one is lactoferrin and another one is Survivin. As I said, it is a multifunctional innate immunity component. It is a single chain 78 kDa iron binding protein. So this iron binds it here. It has the NHC lobes, 2 lobes, and this is the iron element where the atom which binds here. So it is present in the most mammalian exocrine fluids because it is innate immunity component. So it is present as a first line of defense in the saliva, bile, pancreatic, gastric juices, tears, milk, colostrum. And it has immunomodulatory and anti-inflammatory, antifungal, antimicrobial, antiviral, antioxidant properties. When we search the literature, first discovered in 1939 from the bovine milk, cow's milk, the number of its properties have been identified, and it fascinates all the researchers. And it has, the most important thing was with the clinical trials in the Japan on the colon cancer patients, it has gained attention as an anti-cancer chemopreventive and therapeutic. So its oral feeding has proved non-toxic and restores both red and white blood cells which were otherwise depleted by the cancer chemotherapy. On the other hand, Survivin is a protein which is a bona fide therapeutic and diagnostic target because it is overexpressed almost in every tumor. Normal cells, normal differentiated cells lack Survivin. But in cancer, it is a targeted, hot, validated target. As Professor Kanwar has mentioned earlier, that there are a number of ways targeting Survivin. There are the small molecule inhibitors, there are the dominant negative Survivin mutants, and there are the siRNA, but none of them has reached the clinics yet. So there is a clear void in the Survivin inhibitors, which is a clinician's dream because Survivin enhances the radio resistance, the chemo resistance, the metastasis, it is highly expressed by the cancer stem cells. So you must be wondering why to go further if we have to target such a potent anti-tumors as a tumor survival factor, why to go for the milk bioactives? Actually, our research started with on lactoferrin started with Lactopharma Consortium in the University of Auckland, where they were looking for the milk bioactives. And milk has a naturally occurring non-toxic number of proteins, nutritional components, and the most important component is lactoferrin. This protein has got such a famous status in the science world that after every 2 years there is a conference dedicated to protein lactoferrin, and this time it's happening in Japan. This is ages old, centuries old story maybe. So this figure is taken from where they show in the Roman legend, where it is that these are fed by the milk to survive for the healthy life. Studies have proven that this is bovine lactoferrin is safe, so it has been given orally and up to 2,000 milligrams per kg per day, and there is no toxicity effects. And it has been there has been a lot of research based on which the lactoferrin is marketed in over the shelf counters in the health stores because of its properties, and they are using the validated scientific properties of lactoferrin as an antifungal, antibacterial, anti-tumor, and because of its widely reported multifunctional properties and a large number of clinical trials, including in the infants, the pregnant women, which have proven its safety. So the European Food Safety Authority has recently given a green chip to this one for the dietary supplement to be used in food products, infant formulas, and sports medicine. It is the component of the whey protein, which is used for the muscle building and for the cardio ones. So how lactoferrin does all these properties, these actions, these activities? Lactoferrin binds to the mammalian cells through its receptors. It internalizes into the cells through the membrane-bound surface receptors. And as Professor Kaur has already shown this one, so in the gut when it is orally taken, it binds to the receptors and passes the mucosa. It's taken by the immune cells, and then it has its immunomodulatory functions. So similarly, when we did the studies, so when it was fed in the oral through the oral feeding, so we found that the lactoferrin, especially the iron-saturated lactoferrin, because it is a non-binding protein, and being more gut enzyme resistant, digestion resistant, it has the fat protein reach the tumor sites. But this was not sufficient. So to improve this one, Professor Kanwar has patents, and I was a part of at the time with this collaborative research from Fonterra funded research, and they have developed this one as the world first medical food known as Recharge, because it complements the chemotherapy and elevates the patients from the chemotherapy-induced side effects. So coming to this, as it is a citrophilin, so as we lower the pH, this protein releases the iron, and as we increases the pH, the iron binds to the protein. This one is not working, I think it's gone. Sorry. So the iron binding makes it more gut enzyme resistant, and it survives reaches the tumor sites as a whole molecule, which is a good thing because it. Now coming to the Survivin, as I said, it is a bifunctional protein because it has a role in the mitosis, and it is an inhibitor of apoptosis family protein. But it is always, in regards to tumor, it is associated with the poor prognosis, associated with the high expression of the clinical oncology studies. how it works, it works both caspase independent and dependent pathways and mitochondrial membrane permeabilization intrinsic pathway. So using the proof of concept colon cancer, because it is a highly reported second leading cause of death in Australia and other countries, it's emerging with the Western food, Western lifestyle. Even the colon cancer is increasing incidence in the other countries, the developing countries. So conventional treatments have, as I said, they have the harsh drug resistance. So we used lactobacillus. We tried, however, why not using this proof of concept model? Let's look for what happens in the colon cancer. So I will not take over much as this has been a repetition. So we developed the novel alginate coated chitosan calcium phosphate iron saturated lactobovine nanocarrier system to deliver, because initially feeding, so we have seen that there is only a small amount of the protein reaches the tumor. To make it more bioavailable, we prepared the nanocarrier systems, which Professor Kanwar has already explained to you. So it reaches, and the interesting thing we have seen is that after treatment, there is a complete, the mice became complete tumor free when we have fed the colon cancer cells. And in the preventive studies, in the preventive mode, 20 mice didn't develop. We did 5 first, 10, 15, 20. So 20 mice after given, they didn't develop the tumor at all. So they were fed for 2 weeks before, and then they were given the injection, they were looked for. And in the treatment, it brings down, as compared to the Taxol and other injections, it brings down the tumor to the very quickly. And this protein with the nanocarriers actually delivered the protein to the tumor site. And this was the mechanism, because they were non-targeted, so we thought that they were working through the enhanced permeability retention. But what is the mechanism? Why the mice is tumor free? Why the tumors are going? So why there is no recurrence? Even after the second challenge, there is no recurrence of the tumors. So this is what actually we are looking into, because there is, if it is apoptosis, so it could be, because earlier studies have given the information that it targets the Fas ligand, caspase 3, caspase 8. Lactoferrin targets the other BCL BAX2 ratio, but no one has touched Survivin, which was so important. So we hypothesized that lactoferrin targets Survivin, and but before doing, we did some in vitro studies, and this was the lactoferrin, and we looked into that after binding to iron, its immunological properties remain the same, its structure remains the same, it can be identified through the antibodies, and it binds through the receptor mechanism on the cell membrane. So this is what it is binding. This is the iron-free apolactoferrin, the iron-saturated lactoferrin. And we developed the 3D tumor tissue models, which are the spheroids, and it is internalizing very well into the tumor tissues. So lactoferrin binds to a receptor mechanism, receptor mediated, and because in the tumor spheroids, the CD133 is the stem cell marker. So we tested if lactoferrin can target the stem cells in a population, and as you see that lactoferrin, this is the iron lactoferrin, which is highly targeting the stem cells of the CD133 expressing cells. You can see the yellow co-localization of the ones, and the APO is still struggling, but our studies have shown that these two types of the lactoferrins, they target the different receptors. So that could be the possibility. So lactoferrin, iron saturated, is internalizing much faster than the apolactoferrin. This could be the reason it was very effective. So we, through the clonogenic assay, we see that lactoferrin completely abolishes the clonogenic growth of these colon cancer cells. And through the APO process, an XNV assay, we have seen that iron-saturated lactoferrin actually induces the apoptosis, early apoptosis in XNV positive cells. And while the other two proteins, the other two drugs like Taxol, it increases more necrosis also. So that could be bringing a chain of reactions into the tumor site, because once there is a necrosis, there is an inflammation, there is more destruction of the tissue, the neighboring tissue, the normal tissue. And similarly, apo being iron-free, maybe it is also chelating the iron from the milieu of the tumor microenvironment, so it is causing necrosis. But iron-saturated lactoferrin is causing apoptosis, which is what we want. And we then see that lactoferrin, whether it targets survivin in the KK2 cells. So if you look into, they have more than 90% of the stem cells in KK2 cell line. So they express very high levels of this survivin protein, which can be easily seen as the specs, which indicate that it could be the mitochondrial relation, mitochondrial survivin also, but it's cytoplasmic survivin as well as the nuclear survivin. So when we treated the cells with 24 hours with these proteins, we have seen that it down-regulates the survivin expression. And this was seen through the Western blots also. So iron lactoferrin has shown the complete down-regulation of survivin, and while there is a up-regulation of the BAX to VCL ratio, the cytochrome C is released much more, and there is the lactoferrin, and because of the lactoferrin, the caspase 3 is getting up when the survivin is down. So this was a very clear indication, because caspase 3 survivin binds to the caspase 3 and inhibits the apoptosis, because caspase 3 is the final executioner of the apoptosis. So this is what we are clearly seeing: when there is no survivin, there is the caspase 3 expression. Then we not stopped here. We thought that let's see if it is reproducible in the other cancer cell lines. So we did the other cell lines, which are the liver, 2 cell lines of the liver and 1 more colon cancer, and it is more aggressive form of the cancer. So we have seen that it also induces, the treatment also induces that this is a normal cell, but it is making it apoptotic. So there are the apoptosis, and the cell growth is decreased as compared to the untreated cells. And the tunnel assay again proves our findings that it is the lactoferrin treatment, especially the FABLF, the iron saturated, increases the tunnel positive cells, which are the apoptotic cells. And in the liver and colon cancer, again, it is we repeatedly found that it decreases the clonogenic growth of the cells very highly significantly. So we did the apoptosis array, where we have actually looked into the protein expression of the protein profiler array, where we have looked into the 35 type of the molecules which are involved in the apoptosis modulation. And these are the changes with the pro-apoptotic proteins and the anti-apoptotic proteins. So what was very significantly remarkable is the down-regulation of survivin expression and HIF1 alpha, the hypoxia-inducing factor. And it is they have a direct relationship. If 1 alpha is increased, survivin is increased. So it is down-regulating both the molecules and is up-regulating the BAX and the pro-apoptotic SMAC, which binds to the lactoferrin, and also another class of the inhibitor of apoptosis families. And this was repeated in another cell line, again to the different level. So survivin and the hypoxia one was, the HIF1 alpha was again targeted. And because the hypoxia leads to the angiogenesis, there is a VGF, and hypoxia actually leads to the tumor growth. So when there is this was a very interesting finding, because it is targeting hypoxia as well as survivin together. And so that could be the reason, because it is that we don't see any tumor growth. It's not only survivin. And in the cell lines, this is a...
One more minute.
Okay, yeah. In the cell line, there is I'll quickly sum up this one. In the cell lines, when the hypoxia is there are the polygyne cells, cancer cells appear, and they appear more, and these are the cells which are responsible for the recurrence of the tumors. And these cells get highly when there is a drugs. When we treat them with the chemos, these cells get highly. That's why after even the chemotherapy, we get the recurrence. And this protein, the lactoferrin, it targets the polygyne cells, and as well as, so this has given us another indication that this protein has a certain potential which we really look into, and in the survivin targeting, because of the drug treatment, survivin gets upregulated, the polygiant cells get upregulated, the stem cell markers get upregulated because of the chemos, because that is the survival mechanism of the tumor cells. When there is a stress, they want to retaliate back, not to have that. So that's why, and if we give this protein along with the chemotherapy or on its own, it is targeting the tumors and the hypoxia and its related mechanisms. So, and more interestingly, it is not toxic to the normal cells. So that's why it is, that's what we have shown in the feeding trials, and this is showing in the lab studies also. So I will cut my talk, and because we have done in the breast cancer similar findings, and we have done the melanoma studies from the patient cell lines, patient-derived cell lines, and more interestingly, we find that it works through the p53 pathway. So p53 is a tumor suppressor protein. So and it upregulates the p53 pathway, the survivin gets downregulated. So it is targeting survivin through hypoxia, through p53, and through other pathways. So that's the findings so far. And we have just recently published the paper in the this which has been highly accessed. It is just came up 6 weeks ago, and this is the one where we have published the nano formulation in the cancer stem cells, and this was the iron-free differentially modulate. And we think that this can be effectively exploited for future oral biodrug targeting survivin, because it is safe and it gives and it is well tolerated because it is a protein. There is no lactose intolerance. And I acknowledge my team, especially my colleague Professor Kanwar, my associate Professor Jeff Christensen, with whom we worked for the Frontiera in Lectopharma Consortia, where we started the lactoferrin work, Deakin University for all the supporting me with the research, the Cancer Summit Conference Organizing Committee, the grant fundings from Australia Strategic India Strategic Research Fund, NHMRC, Frontiera, New Zealand. So, and these, of course, these are my students who have done work on the different models. So thanks to them, thanks for their wonderful work. They have presented their data, and thanks to you all. Any questions?
Thank you very much, Professor Kanwar, and we appreciate the topic. Any question or comments?
So, have you tried looking simultaneously at CD133 and HIF alpha?
Yes, I didn't present the data. So because whenever there is we have done in the tumor spheroid models, when we grow the tumor spheroid models, there is a central core which is necrotic, and where the hypoxia is there. And then we stain the cells on the periphery of the cells. And it has come up a very beautiful figure, actually very beautiful. We have seen that there is the FCAM positive, CD133 positive, CD44 positive cells are there. And we have seen, because the lactoferrin, when we treat with the lactoferrin, the outer core dies so quickly. So,
Yeah.
Sure.
You mentioned about how lactoferrin can suppress the survival through the p53 dependent pathway. Have you tried to test whether the p53 mutant cancer
Yes.
cells also affect?
Yes. So we have done to the actually different cell lines, because we have done in the breast cancer, which we published recently. MSF7 is a p53 wild type. MDA-MB2 is 231. Similarly, with our these patient cell lines, this is one of the figures from the patient where p53 dependent pathway. So we have the drug resistant cell line and the two patient cell lines where we have worked, and we have silenced the p53 to see if there is the one. And this one shows that inactivation of the p53 using the siRNA actually didn't lead to apoptosis in any of the melanoma cell lines tested. So this is, we are looking for the p53 pathway. And where there is a wild type p53 pathway, it works through the p53 pathway. If it is not there, then it works through the alternate pathway, the p10 pathway. whichever work we have done in the prostate cancer.
Okay. Good. All right. Thank you again.